Hello everyone. So today is microbiology class. So in this we will learn about bacterial cell structure. So we continue with the bacterial cell structure. Here you can see a picture of bacterial cell. In this bacterial cell you can see there are different number of structures which is labeled in that structure. Here you can see there are cytoplasm, pilus, flagellum, ribosomes, DNA, plasma membrane, cell wall, and the capsule. So we are starting describing the structure from the outside. So here you can see the outer layer which is present in the dark blue color. So you can see the dark blue color. This is the outermost layer of the bacteria, and this structure is known as capsule. So the outermost layer is capsule. So in some bacteria this capsule is present, means this capsule layer is present, and in some bacteria this capsule layer is not present. So it is not necessary that all bacterial cell is having that capsule layer. So if the bacteria who is having that capsule layer, so means that bacteria is having a more virulence power as compared to the bacteria who is not having this capsule layer. So now we come to the middle one layer, which is present after the dark blue color, which is present in like little white color. So you can see that layer is known as a cell wall. So this is a middle one layer. So this layer is known as a cell wall. So this cell wall is basically indicating and responsible for the shape of the bacteria. So this is a rigid layer, so that's why it is maintaining the shape of the bacterial cell. After the cell wall, you can see this is the next layer, the innermost layer you can see, which is indicated by the plasma membrane. So you can see here that it is labeled, so that is a plasma layer. So this is the innermost layer of the bacterial cell. So basically the bacterial cell is made up of a three layer. The outermost layer is capsule, and the middle one is cell wall, and the innermost layer, that is plasma membrane. So now we come to beneath the plasma membrane. You can see inside this bacterial cell there is one fluid, that is transparent fluid, and that fluid is known as cytoplasm. So this cytoplasm is having a number of material and the structure which is present inside the bacterial cell. So you can see here in this structure, the green one structure which is present in a bunching form. So that green color is indicating that is DNA material, so which is a basically genetic material. So inside the cytoplasm, in between, you can see that is a green material is known as a genetic material, which is known as a nucleoid. So if we compare with the eukaryote cells and the prokaryote cells, so basically this bacterial cells are the prokaryote cells, and if we're talking about the human cells, so the human cells are the eukaryote cells. So the one difference between the eukaryote cells and the prokaryote cells is related to this genetic material. So means in the prokaryote cells, means in the bacterial cells, the genetic material is not covered by any nuclear membrane. So that's why this structure is known as a nucleoid. If we are talking about for the eukaryote cells, means that genetic material is covered by a nuclear membrane. So that is known as a nucleus. So this is the basic difference between the eukaryotes and the prokaryote cells. Now we come to the next structure which is present inside the cytoplasm. You can see here the black colored dots are present. So that black dots are present is known as a ribosome. So these ribosomes are basically structure which is responsible for the protein synthesis. So protein synthesis is necessary for the bacteria because for the DNA replication the protein is necessary. So this protein synthesis will be only taken place by the ribosome only. So ribosome, which is basically helping in the protein synthesis, and with the help of the protein the DNA material will able to replicate, and that will also lead to the division of a number of offsprings of bacteria. Now you can see inside this cytoplasm also we are having some floating materials which is present in a disc-like shape. So this disc-like shape material which is known as a plasmid. So plasmid is basically a genetic material. So this genetic material which is not present in the bunch of this DNA material, these material which are floating inside the cytoplasm. So this floating genetic material is not present in all bacteria. In certain bacteria these are present. So this type of a floating genetic material is known as plasmid, or also it can be calling as a episome. So now we come to the next structure. You can see here the green short thread-like structure you can see, which is present all over the structure of bacteria. So this green-like structure is known as a pilus. So pilus is basically it is a thread-like structure which is responsible for the transfer of genetic material, or you can say it is a organ of a reproduction, which is basically responsible in a bacterial cells. Now you can see there is a one tail, a brown color tail is there. So this is a long tail which is known as a flagella. So this flagella is a structure, a long thread structure which is longer than the pilus. So the function of that flagella is for the locomotion. So means the bacteria is able to move from one place to another place with the help of this flagella. So basically this flagella is an organ for a locomotion. So this bacterial cells will be able to move with the help of this flagella. So we can identify the bacteria, whether the bacteria is motile or non-motile, with the help of the presence of this flagella structure. So we have seen this structure, and we just described the all details about the structure of the bacterial cells. So now we come to one by one with these structures. Now first, already told, the bacteria is made up of a three layers. So first outer layer is already told, that is a capsule which is present in some bacteria, and in some bacteria it is not present. So outer layer we can say that is a rigid cell wall, and second one is a plasma membrane. So after the plasma membrane you can see it is having cytoplasm and cytoplasm inclusion. So in the cytoplasm inclusion means inside the cytoplasm we are having number of a structure like mesosome, ribosome, inclusion, granules. Now cell wall. So cell wall is a tough and rigid structure which is present surrounding the bacterial cell. So the outer one layer, that is a cell wall, which is a very rigid because it is maintaining the shape of the bacteria, and it is made up of a peptidoglycan. Now second is cytoplasmic membrane. So this is a innermost layer which is present after the cell wall. So this membrane is basically separating the cytoplasm, that is a transparent fluid which is separating that cytoplasm from the cell wall. Third one we are having, that is cytoplasm. So cytoplasm, already told, it is a transparent fluid which is present inside the cell envelope, and it is having a number of structure like ribosome, mesosomes, vacuoles. So first is ribosome. So already told, it is a center of protein synthesis. So it is helpful for the protein synthesis. If the protein is synthesizing inside the bacterial cell, then only the DNA material will be able to replicate inside the bacteria. Next you are having intracytoplasmic inclusion. So inside the cytoplasm we are also having a small bodies. So these inclusion bodies are known as intracytoplasmic inclusion body, and the function of this basically is a source of stored energy. So these bodies are storing the energy. It means whenever the bacteria is not able to take the food and is not able to find out the food, so in that condition the bacteria can use this stored energy, and this stored energy will be available in these type of cytoplasmic inclusion body. Next is mesosome structure. So mesosome structure is basically it is a projected structure or multilaminated, or we can say it is a finger-like structure which is originating from a plasma membrane. So basically this is a center of respiration of the bacteria. So the cellular respiration of bacteria is done by the mesosome. And also one point is, if we are differentiating between the eukaryotes and the prokaryote cells, so in the prokaryotes, means in the bacterial cells, the mesosome is responsible for the respiration. But if we are talking about the eukaryote cells like the human cell. So in the human cell, there is a structure inside the cell, that is mitochondria. So mitochondria is responsible for the cellular respiration in the eukaryote. So this is also the second difference between the eukaryotes and the prokaryote cells. Now next structure is the bacterial nucleus, which is already shown in the picture. That is the middle one structure which was showing in a green color. So that is the genetic material. So that genetic material which is present in a cluster form and it is covered by a nuclear membrane. So that structure is known as a nucleus. And second is plasmid. So some bacteria is having a extra nuclear genetic material which is not present inside the nucleus, which is basically floating inside the cytoplasm. So that type of structure and that type of extra nuclear genetic material is known as plasmid. So this plasmid is not essential for the life, because already told कि in some bacteria this type of structure is present. So the bacteria who is having this plasmid structure means the extra nuclear genetic material. So that type of bacteria is having a more virulence power because they have a drug resistance and they are having a more toxigenicity as compared to those bacteria who are not having this extra nuclear genetic material. Now we come to next structure, that is bacterial capsule. So bacterial capsule, the same, it is the outermost layer which is present in certain bacteria. So with the bacteria which is surrounded by the capsule, these bacteria are having more virulence power. So basically, what are the function of bacterial cell? It is acting as a protective covering for the bacteria against the antibacterial substance. Means the we are using to kill that type of bacteria, so it will take a time to kill that type of bacteria because the capsule is working and acting as a protective covering for the bacteria. So that's why these bacteria are having more virulence power. Now come to next structure, that is fimbriae and pili. So pili already shown in the picture. There is a small thread like structures which is known as a pili. So this is also known as a fimbria. So these are projecting from the cell surfaces, and also told ki the function of the pili is transfer of genetic material, means they are able to transfer the genetic material and they are helpful in the reproduction. So they are helpful in the reproductions of bacteria from one bacteria to the number of offsprings bacteria. Next structure is we are having, that is flagella. So flagella is already told it is a long thread-like structure than the pili. So which is protruding from the cell wall. So basically the function of the flagella is for the locomotion, means with the help of the flagella the bacteria is able to move. So these type of bacteria are motile. Bacterial cell structure. So this is all. This is again the reference from which I have taken this topic. So basically we have studied and discussed about the bacterial cell structure.